Good evening. Welcome to our GeCIP seminar and Queen Mary University of London seminar series. We've got two fantastic speakers this evening, as usual. We're starting with Patrick Chinnery. He is a professor of neurology and head of the Department of Clinical Neurosciences at the University of Cambridge. He is known internationally for his work on mitochondrial disorders, and he's an NIHR senior investigator. He jointly chairs the NIHR Bioresource for Translational Research in Common and Rare Diseases. He's chair of the Medical Research Council, Neurosciences and Mental Health Board, and most importantly, he is co-lead of the Neurology GeCIP domain. So welcome this evening. Thank you.
I was a little bit unsettled as you were looking on your phone for some information about me. I'm not quite sure what you were going to find.
I know who you are, but the words get muddled.
Yeah. Great. I just want to swap computers if that's okay, so the transitions work. Thank you very much for inviting me. It's a pleasure to be here and to talk to you about, if you like, the other genome. But I think what I'd like to do is give you a bit of background, place it in the context of some work that we've been doing in my lab to understand about how mitochondrial diseases arise, and particularly mitochondrial DNA diseases arise, and then move on to some illustrations of how working with the NIHR Bioresource and GEL dataset is really helping us to push our understanding of these interesting biological mechanisms that are relevant for disease. And I think it's a nice example to illustrate how the dataset that we've all been involved in generating is rich for research purposes, and that by mining it you can discover things about biology, about disease, that were not the primary intent at all. So it's the power of the genomic resource. And I'm going to talk to you about mitochondria, which, to remind you, are these organelles that are present inside all of our cells, and they're important because they're the main source of adenosine triphosphate, which is the energy source that the cell uses for all active processes. I'm going to talk to you about primary genetic disorders of the process that makes ATP, which is oxidative phosphorylation. Now, oxidative phosphorylation is fundamentally important for life, and consequently there are other contexts that are not primarily genetic where dysfunction of mitochondria can cause diseases. And so there's a whole world out there of people exploring mitochondrial mechanisms in sporadic diseases and genetic diseases that are not primarily mitochondrial. But I'm not really going to talk about those today, although it may well be that this data set's useful to help us understand some of that. So I became interested in this as a junior neurology trainee in the northeast of England at the time, and I became interested because it was the time in the field where these disorders were just starting to be described for the first time. Multi-system diseases that often presented with neurological features like the eye movement disorder and weak eye muscles at the top there, dysfunction of the brain, causing diseases that looked a bit like Parkinson's disease, clumsiness, which neurologists call ataxia, deafness, myopathy, damage to the nerves in the arms and the legs, and also features outside the nervous system, such as in the gut, or affecting the endocrine hormone systems, affecting the liver and the heart, particularly in children. And the thing that fascinated me and some of the members of the audience here, like Shamima Rahman, who's come along, the thing that fascinates us is these different clinical features with different people having different combinations of these, which is one of the reasons, I think, why these genetic diseases were missed for several decades. And by studying families with rare inherited mitochondrial disorders, we've learnt a lot about how mitochondria are put together, because these families, these patients, have lesions, if you like, in the system, and when we characterise those lesions, we can work out the key parts of the machinery that are essential for normal mitochondrial function. And central to this is this group of 100 proteins arranged into 5 complexes that form the respiratory chain that make ATP. And this respiratory chain is synthesized from 2 different genomes. From mitochondrial DNA, which is a circle, and there are many copies of this circular genome that are present inside the mitochondria. And this genome goes on to contribute to protein components of several of these respiratory chain complexes. But the vast majority of the mitochondrial compartments or components come from the nuclear genome, estimated to be about 1,500, such as most of the proteins that make the respiratory chain, and some chaperones that allow the complicated machine to be assembled on the inner mitochondrial membrane, and the proteins that allow you to replicate, copy your mitochondrial DNA and maintain it, and also the transcription, translational machinery, which allow the mitochondrion to make the proteins derived from mitochondrial genome inside the matrix. So all that comes from the nuclear DNA. And so it's not surprising, therefore, that when you think about genetic disorders of this system, it can arise either from mutations of the mitochondrial genome or the nuclear genome. And if we just look at the way this field's unfolded over the last 25 or so years on this slide, this is a summary of the number of different genes that have been identified in patients with mitochondrial diseases from 1988, when the first ones were found in red here, mitochondrial DNA, through to the back end of last year when we updated this. And what you'll see is in the early stages the focus was on mitochondrial DNA, but over the last 15 years this has transformed, initially with linkage studies and candidate gene work, and then onto massively parallel sequencing. And several of us are involved in trying to discover new mitochondrial disease genes which will help us understand the system more and provide a diagnosis to our families. And the mitochondrial GeCIP, which Shamima co-leads that, is involved in harnessing the resource of patients that have been recruited with unknown causes in the GEL dataset to find these new disease genes. But I'm not going to talk about that today. Sorry, my computer's doing a weird thing. Let me just shut this thing down. So, colleagues in Newcastle, Oxford and London have been really central to providing mitochondrial disease diagnostic services for the last 2 decades, and that allows you to construct some crude epidemiology based upon the patients that have come out of the woodwork. And I was involved in a number of studies, most recently updated by Grainne Gorman, shown on this slide, which describes the prevalence of these disorders in adults. And the take-home message is that they, as a group, they affect about 1 in 8,000. And when you look at the mitochondrial DNA basis, in adults at least, about 2/3 of the patients we see have a mutation of mitochondrial DNA causing a mitochondrial disease presenting in adult life. So clearly this is an important question that I'm trying to answer in my lab and through collaborations to try and cast light on the question, where do these mitochondrial DNA disorders come from? So let's just step away from the nuclear genome and remind ourselves a bit about mitochondrial genetics. So the molecule resembles a bacterial genome. It's small, it's 16,500 bases. We think it just codes for the proteins in the respiratory chain. And in between those different coloured genes, which are the protein coding genes, there are some ribosomal genes, and the yellow bits are the transfer RNA genes that are required to make the proteins inside the matrix. And the prevailing view is that this genome is essentially maternally inherited, although there may be some exceptionally rare exceptions to that. For all intents and purposes, it's a maternally inherited genome. And because there are many copies of this present inside each cell, you get a rather different situation to the nuclear genome. You can get, if there's a mutation present, it can affect a varying proportion of the molecules. And we know from studying cell lines from patients and single cell studies that generally for pathogenic mutations the cells can tolerate quite a high proportion of the mutated genome. And it's only when they go above a critical threshold that this affects the synthesis of adenosine triphosphate and you get a biochemical defect. So the 3 differences are multi-copy, so there may be thousands in a cell. You've got this situation of mixed genomes, which is called heteroplasmy, and there's this threshold effect, implying that below the threshold, the mutations may not be doing much. And for the most part of my earlier career, the view was that this mixed population of mitochondrial DNA was actually very rare, maybe found in 1 in 25 people, based on the technology that we had at the time. But Brendan Payne, who worked with me a few years ago, was one of the first people to set up deep resequencing in our lab. This was done in 4 or 5 labs around the world at the same time. And it allowed us to look at the mitochondrial genome in unprecedented depth, and in particular to try and see whether or not, using a very sensitive method, we might find that heteroplasmy was more common than you might think. And of course the answer is it is. What this graph shows you is it shows you the relationship, the level of a heteroplasmic variant, a single base pair variant in mitochondrial DNA in one family member compared to another maternal family member. And each dot is a different family, and it's actually a different variant too in each family. And the take-home message from this slide is that if you get down to about the 1% level, so 1 in 100 mitochondrial DNA molecules have a variant, it's almost universal. We're all carrying these heteroplasmic variants at extremely low levels that we've not picked up before. So maybe this is telling us something about where these diseases come from. To do that, of course, and based on what I showed you in the last slide, these levels of 1% are unlikely, based on current knowledge, to have a phenotypic effect. So the level's got to change if it's going to cause disease, and there are a variety of ways that this can happen. They're shown on this slide. At the top is a process called vegetative segregation, and the basic idea is that if you've got a cell with a heteroplasmy, when the cell divides into two daughter cells, by chance, you might pass different proportions of the mutated and wild-type genomes to the daughter cells, and the same thing might happen again. So you can see, with a few cell divisions, you might go from a mixed situation to fixation or very high levels. So heteroplasmy can change when cells divide. And it's more complicated than that, actually, because even in cells that don't divide, the level of heteroplasmy can change. And it can change because the genome is replicating independently of the cell cycle. It's called relaxed replication. And so each, over time in a cell, like a neuron or muscle, then genomes are being destroyed and replicated over time. And this has a half-life of about 10 days in rats, so it's quite a lot of this going on. And it might explain why there's a high mutation rate. But the reason why it's important here is because you can see that you might by chance replicate mutated genomes rather than wild type over time, and that if that happened again and again by chance or because there was a selective process, you can see how the heteroplasmy might change during life in people. And we see this in our patients in muscle biopsies. So these in principle are ways that this very low level of heteroplasmy can change. So let's get on to our clinical problem, which is how these diseases arise. And this is a typical presentation of a child who developed a neurological disorder in infancy called Lee syndrome, with the characteristic findings on the brain scan with these abnormalities here in the basal ganglia. And the child had this mitochondrial DNA mutation. And here is the child here, and we made the diagnosis by studying his mitochondria in muscle. So we measured the level of this heteroplasmic mutation in muscle, and it was 98%. And when you trace back through the maternal line, you'll see that different family members have different levels of the mutation, even within this tiny family. And this is something that's fascinated me for probably far too long. It's something that my wet lab focuses on, and it's something that the GEL data set is trying to help us understand. So just to frame the biology, if we're going to understand how these mutations are being passed from one generation to the next down the female line, we need to think about germ cells. And we need to think particularly about female germ cells in this context, and most importantly, the process by which the primary oocytes are made. Just to remind you, these are the germ cells that are made during the development of a woman, when she's in utero, when she's a fetus in utero, during the first trimester of life, of pregnancy. These oocytes are formed, and stuff that we've done in our lab and other people in Canada have shown that if you measure the level of heteroplasmy in primary oocytes from patients actually, and also from mice, you can see a range of heteroplasmy values which matches what happens in the next generation. So the point is the die is cast by the time the primary oocytes are formed. So to understand how this happens, we need to go back in development to these things called primordial germ cells, and possibly even further back to the blastocyst and the fertilized zygote, because if heteroplasmy is going to change when a mother passes this to a child, we know from mouse studies that this is when it's likely, this is when the change is likely to be happening. And we've been studying the biology of this in human embryos for the last few years. This is some data from healthy human embryos between weeks 3.5 post-conception and week 7, stained with something called tissue non-specific alkaline phosphatase, which is brown. You can see the germ cells first starting to appear 3.5 weeks after conception. This is the gut creeping down the gut mesentery, and within a few weeks there are the ovaries stuffed full of these early germ cells, proliferating still and forming these early germ cells. And we spent a fair while trying to isolate these to study their mitochondria, and Vasilios, who works in my group for about 5 years, mastered this technique of isolating germ cells using fluorescent sorting methods and cell surface markers. And it allowed us to look at this process in humans for the first time. And just to summarise the data before we get onto the gel stuff, this is what we found. So we took these human embryos that had been donated for research, and we fact sorted them at different stages in life during development, and isolated these early germ cells. And here's one. It's about 5 microns across, and the mitochondria are stained in green, and there are about 300 or so in each one of these germ cells. And then we took the germ cells and deep sequenced them 1,000 to 2,000 fold to see whether what we'd seen in our families was actually present in the germ cells. And the data is summarised here. This is the genome, mitochondrial genome. There's a long linear strip, and these are different heteroplasmic variants found in single primordial germ cells isolated from healthy human embryos. And what it tells us is that they're there, that these healthy germ cells are carrying mitochondrial DNA variants with varying levels of heteroplasmy. Some of them look pathogenic and some of them don't. So the question is, how does this change? How is it that these low levels of heteroplasmy that we find everywhere can go up to high levels and cause diseases? And there's been a hypothesis that was first really proposed based on work done in cows, heteroplasmic cows in the 1980s, where Olivio, Holsworth and Lapis looked at these cows and saw that heteroplasmies in cows could become fixed on the variant, or a new variant, within 2 or 5 generations, really fast in terms of population genetics, from one allele through to another. And they came up with this idea that there was a genetic bottleneck during the transmission of mitochondrial DNA, illustrated here. The idea being that if you've got heteroplasmy and you only take a small proportion of the heteroplasmy each time you make a germ cell, you'll get varying proportions in each germ cell, and that these will be passed on to the next generation. And because you get varying proportions, you can get varying levels of heteroplasmy that are inherited down one generation. So we've tried to characterise this in humans by looking at our human primordial germ cells from week 3.5 to week 7 and measuring the amount of mitochondrial DNA in each cell, and you'll see the amount there is about 1,200 or 1,300, which is a lot less than an oocyte. Human oocytes contain 500,000 or so copies of mitochondrial DNA, so it's clearly gone down at some point. To nail it, we work with Dusko Ilic at King's, who generates, for research purposes, human pre-implantation embryos, allowing us to look at things like the trophectoderm and the inner cell mass and measure mitochondrial DNA levels, and we found that they had more than these early germ cells. So the levels clearly come down, and the key question for us was what was happening in between. Could we show there was a really tight genetic bottleneck? Challenging thing to do, because it's not possible to get those samples from clinical samples, because these are taken from embryos that are donated from people who are having terminations in the hospital, and the earliest we can really get practically is about 3.5 weeks. So the only way of looking at this is in a stem cell system. And Azim Surani in Cambridge, who I've worked with for years, has this system where he can make things that are rather like germ cells in vitro from embryonic stem cells by adding a cocktail of cytokines. And essentially what this shows you is that over about 7 days you can turn these human embryonic stem cells into something that resembles the germ line, because they express the relevant markers. And so we've studied these and measured the amount of mitochondrial DNA in them. And the pink colour are the female ones, and the level in the human primordial germ cell-like cells in vitro drops down to about 400 copies of mitochondrial DNA. So, big change. you've got this fertilised zygote with 500,000 copies, goes right down to a few hundred copies, and then it starts to creep up again as the germ cells proliferate, migrate, and form the ovaries. So I think this tells us there is a bottleneck, and we can say that there's about 4 to 5 molecules per cell. Now we're on to the gel stuff. Okay, so this is the concept that when you pass these things on, there's a decrease in mitochondrial DNA content. You can see shifts in heteroplasmy level, and that might explain what we're seeing in families like this one, which I showed you a while ago. The question is, what's happening in the population at large, given that all of us are carrying these mitochondrial DNA mutations? Is this happening all the time? And it was really based on this idea that we first started to look at whole human genome sequence data derived from the NIHR bioresource and pilot study of Genomics England pilot, which I was involved in when I was based in Newcastle, and the bioresource more so down in Cambridge. So how does this work? Well, when you sequence a whole human genome, you create multiple reads of the genome, and typically you would produce, for diagnostic purposes, something between 30 and 60-fold coverage of the nuclear genome. But of course, there's also mitochondrial DNA in the cell, and when you just extract nucleic acids, you get it with the nuclear genome. In fact, it accounts for about 1% of the total nucleic acid amount in a cell mass, because there are lots of copies of it. And so you not only get a read depth for the nuclear genome, but you also get a read depth for the mitochondrial genome. And because there are many, many more copies of this in each cell, typically in a lymphocyte there might be 500 copies for every cell nucleus, you get a proportionate coverage of the mitochondrial genome. And this shows you for the different early sequencing runs, based on the read length, what the coverage for mitochondrial DNA actually was in this early data set. It's in the region of 1 to 2,000 fold coverage, which allows you to start looking for variants which are rare, down, we think, to about the 1-2% level. And so we've studied this data, and this is very much work in progress, but I'll share with you some of the findings that we've come across. And of course, I didn't do any of this. This was all done by Wei Wei, who sat there, and Ernest Tureau, who's helped us considerably with the statistical interpretation. And what Wei did was she extracted the mitochondrial DNA sequence from 1,500 or so mother-child pairs in this gel pilot data set, also linked to the NIHR bioresource, and looked for heteroplasmy. And with a threshold of 2%, she found that half of the individuals, approximately, harboured heteroplasmic mitochondrial DNA variants. And all of that's summarised here in this cartoon. So here's the mitochondrial genome from 0 to 16.5 kilobases. And what these arrows are heteroplasmic mitochondrial DNA variants that were found in an individual in these 1,500 mother-child pairs. And the arrows and the colours mean something. So the blue ones are ones that are transmitted. In other words, they're present in the mother and the child. And the orange ones are ones that are lost. In other words, we can detect them above the threshold in the mother, but not in the child. And the pink ones are de novo. In other words, we can detect them in one of the children, but they're not present in the mother based on our detection threshold that we use. So we can start to do interesting things with this. It's the biggest data set by an order of magnitude of about 100-fold, and we can start to look at behaviour during this transmission process. For example, we can look at the mutation rate, the de novo mutation rate in unprecedented detail. And what this shows you is it shows you a graphical representation of the different mitochondrial DNA genes. It's a bit confusing to the aficionados. It's going in the opposite direction to normal. But the distance from the centre is the mutation rate, or 1 over log 2 of transform mutation rate, based upon 477 de novo variants that we saw in the children that were not present in the mother. And you can see that certain bits of the genome, like this non-coding region, have a very high mutation rate. That was known. But we're now able to resolve differences in mutation rate, which are per nucleotide, across different regions of the mitochondrial DNA, and it's going to be interesting to see how that maps to what we see in the clinic, for example. We can also look at the data this way. I'm nearly finished, I promise you. So on the left, this is one way of demonstrating heteroplasmy in a population that you're looking at. What it is a cumulative distribution of heteroplasmy. So the heteroplasmy from 0 to 100% is on the bottom, and this is just a cumulative distribution of those heteroplasmies in all of the individuals we looked at. And it's subdivided based upon different parts of the genome. So we separated out the non-coding, synonymous, non-synonymous, ribosomal RNA genes, transfer RNA genes. And what we're starting to see when we've got a data set of this size is there are differences between different parts of the genome. And what this tells me is that the non-synonymous variants are behaving in a different way, as indeed are the ribosomal RNA genes. There's some scent that there's selection going on in the population for and against certain bits of mitochondrial DNA. And you can look at it during transmission. You can see whether or not heteroplasmies are likely to be transmitted or lost, and what the proportion of them is. And you'll see for some of these, like the ribosomal RNA genes, there's a tendency to lose heteroplasmies, but for other bits of the genome, like the non-coding D-loop, there's a tendency to transmit heteroplasmies. So we're starting to get some granularity on how the genome is evolving in real time. And just the final message I'd like to give you is this. This is a map of the world, obviously, and what it shows you is that the migrations of humans out of Africa 200,000 years ago around the world, which in part has been deduced by studying mitochondrial DNA variants by anthropologists, population geneticists over 2 or 3 decades. And what you can gather from this picture is that different parts of the world have different characteristic patterns of polymorphisms of mitochondrial DNA. And we've looked at these in great detail actually before GEL through the NCBI data set where there are 30,000 or so of these. And what it allows us to do is get a flavour for what mitochondrial DNA variants are known and are out there and what ones are not known and are not out there. And you'll see from this diagram, which is the mitochondrial genome, in different parts of the world here, these are these different haplogroup definitions, that these are not evenly distributed. So the genome seems to like some variants and not others. And we can see the signature of this in the GEL data set. I'll just take you through this to the last data slide. What this shows you in these 1,500 mother-child pairs is the number of heteroplasmies we saw, which were previously known in the human population, in our 30,000 NCBI variants, and novel ones which had never been seen before. And we've split it up based on whether the blue ones are transmitted, gold lost, or pink de novo. And what this tells me is that the known variants and the novel variants are behaving differently in the germline, in these trios. And in particular, the novel variants seem more likely to be lost, statistically significantly so, than transmitted, implying that this selective process is actually happening in real time. I have one slide which I'm going to go over. I'm going to summarise. So what we found is we found that heteroplasmy is universal. I suspect if you did even deeper sequencing, we'd find heteroplasmy in everybody. There's a germline bottleneck, and that leads to changes in heteroplasmy that can cause diseases like the patient I described. We think there's a signature of some selective forces, which we've also been studying in the wet lab. And this same process also influences whether these heteroplasmies become fixed or homoplasmic polymorphisms in the population. And there are clearly some suggestion of some signatures of selection going on there. So where does this leave us? Well, practically, I think that's very interesting biology. It's giving us insight into the origins of these diseases. But I also think it's really helpful in terms of our diagnostics, because what this is telling us is that whole genome sequencing is incredibly powerful. Not only is it giving you complete scrutiny, almost complete scrutiny of the nuclear genome, but you can look at the mitochondrial DNA too. And what this donut shows you is it shows you, based upon the epidemiological studies, the proportion of families you might diagnose in principle with a single whole genome sequence. And the answer is it's the vast majority. Over 95% based upon what's known about these diseases could be diagnosed from a whole genome sequence. And I'll stop. Acknowledge all the people that have worked with me, particularly all the people that recruited to the NHR Bioresource Genomics England, and to Wei Wei and Ernest Turo, who did the work that underpinned the latter part of the talk. Thank you very much.
Okay, so now we are switching from rare disease to cancer, and we've got Stephanie Greville-Heygate, who's a specialist registrar in clinical genetics for the Wessex Clinical Genetics Service, and has a special interest in cancer genetics and developing the interface between oncology and genetics. Stephanie has completed the master's degree in genomic medicine at Southampton, and is now working towards a medical doctorate under the supervision of Diana Eccles and the Department of Cancer Sciences at the University of Southampton. She's got a Genomics England Research and Innovation Fellowship and is a member of the Hereditary Cancer and Breast Cancer GeCIPs. So welcome. Thank you very much. Thank you.
I think that's on. Right. So thank you very much for the opportunity to speak tonight and also for the introduction. So this evening what I would like to do is to provide a whistle-stop overview of the current climate for genetic testing in the context of hereditary breast cancer. I'd like to give an overview to my research project using cancer phenotypes to improve cancer susceptibility gene classification. I'd also then like to discuss some of my experience of using the research environment and also some initial results. So, the etiology of breast cancer is complex, and many different factors will contribute to somebody's absolute lifetime risk, including genetic factors and environmental exposures. But approximately only 5 to 10% of breast cancer can be attributable to a stronger single genetic factor which is passed down through the generations. Historically, we've often thought about those single stronger factors in relation to their contribution to cancer risk. So, for example, genes such as BRCA1, BRCA2, TP53 being high risk, genes such as CHEK2 and ATM being moderate risk, and PALB2 sitting somewhere in between, depending upon somebody's family history. So historically, genetic testing for a heritable cancer susceptibility has focused very much on single gene testing in a targeted manner, often for the high-risk genes such as BRCA1 and BRCA2. But in advances in next-generation sequencing technology, a reduction in cost, and also the implementation of work such as the 100,000 Genomes Project, is really facilitating this drive towards the mainstream implementation of cancer genomic technology in routine clinical care to identify both those high-risk gene carriers but also some of the more moderate-risk gene carriers. And this is an exciting vision for the future. But within that, we've got to recognise that to truly harness the power of this technology, we've got to think, what is the clinical utility of this information? So how can we use that to improve the care of the patients that we see every day? But also, how do we interpret it correctly, and what is the cost of that? So, in terms of clinical utility, in terms of the higher risk genes, NICE now have produced clear guidance in terms of how we should manage patients with high risk genes such as BRCA1 and BRCA2 in terms of screening and surgery. But also increasingly we're being able to understand this genetic information in terms of healthcare planning and how best to manage patients moving forwards. And so, whilst we can see there's great potential for clinical benefit through the utilisation of these broader-based genomic technologies, we've also got to recognise there's a potential risk of harm through the identification of variants. And a variant is when there is insufficient evidence to know whether that alteration is truly the cause of somebody's cancer. And it creates the potential for harm through sort of incorrect interpretation, which may result in inappropriate clinical intervention. So we know if we perform targeted genetic testing in a gene that we understand, such as BRCA1 or BRCA2, in a well pre-selected population, the risk of identifying a variant is about 3%. However, if we move into a more mainstream approach, the rate of finding a variant is probably the same, around 3%, maybe a little bit higher, but the difficulty comes when there's no family history to help to interpret that. And we know that the more genes we test, the higher the rate of variance becomes, with actually only a minimal increase in actionable risk. So, we know now, or we're starting to understand for genes like BRCA, that actually tumour histopathology can be a good predictor of germline genotype. So BRCA1 tumours are classically triple negative, so that means HER2, estrogen and progesterone receptor negative. Because of this fact, we now recognise that in women who have a triple negative tumour under the age of 50 years, we will offer BRCA testing irrespective of family history. But also, if we find a variant of uncertain significance in BRCA1, it is 4 times more likely to be pathogenic if they have a triple negative tumour. So, I think what I've shown there is that for well understood genes like BRCA1 and BRCA2, we've got good clinical utility and we've got increasing evidence to help with interpretation. But as we're moving towards more broad-based testing of genes that we understand less well, those questions about what is the clinical utility and how do we interpret it correctly still prevail, with the patient at the centre of that. And that leads onto the research question for my project, which has been, do PALB, CHEK2, ATM and TP53 gene carriers develop tumours that have a characteristic phenotype which can help with variant interpretation? And actually, how does germline genotype influence tumour biology and also patient outcome? Okay, so participants for my work have been derived from 2 studies. So firstly, the 100,000 Genomes Project. And the 100,000 Genomes participants have come from 2 domains: rare disease familial breast cancer, and recruitment to this domain required a diagnosis of a primary breast cancer under the age of 50 years, with 3 affected family members with an average age of diagnosis under 60, or a Manchester score greater than or equal to 22. I also had a second cohort, which were those individuals recruited under sporadic breast cancer. And finally, I've also got participants who've been derived from the POSH study. And this was a study which was established to better understand the genetics of breast cancer in women who've been diagnosed with primary invasive breast cancer under the age of 40 years. And I think the take-home from this is actually that I've got participants from 3 distinct cohorts. So from the 100,000 Genomes Project, I have a familial cohort. From POSH, an under-40s cohort, and also from the 100,000 Genomes Project, an unselected cohort. And based upon the sequences that I've been able to access from the research environment, that leaves just short of 5,000 genomes available. So, the timeline for my work has firstly been identifying participants. It's then been devising a bioinformatics strategy to analyse the genomic data and identify variants. It's then been manual curation of those variants and clinical correlation. So I'm now going to focus a little bit on what I've been doing within the research environment. So when you access the research environment, first of all, to be able to access the genomic data, you need to identify participants. So each participant is listed there with a plate key and an identifier. And you can combine that information to create a file path to access the genomic data. So first of all, you have to create a file path for each of the participants of interest, which was done through command line within the Linux terminal. So it results in a text document which looks a little bit like this. For the 870 available genomes within familial cancer and the 1,339 genomes available within sporadic cancer. So that took quite a long time. So next, I needed to interrogate the genomic data. So a whole genome sequence will generate between 3 and 4 million single nucleotide variants per genome, which is a huge number. And so when the annotated VCFs come back, you need a strategy to be able to filter that data to try and identify variants of interest. And so I have utilised 2 scripts within the research environment. So the first script was a shell script, and I established that to filter to identify coding sequence variants within the genes of interest, and also recognised founder mutations within the genes of interest. So my script looked a little bit like this, and I think really what it's showing you is within that script there are different components. So there's components to filter on the chromosome of interest within the region of interest, to look at the gene, but also to really filter out sections which you're not interested in, like regulatory region elements, downstream gene elements. But I then utilised a second script, which was a bash script, which was actually produced by the GEL team. But this time, rather than trying to pull out coding sequence variants, I've filtered on terms of interest. So whether the annotation said that there was a stop gain variant, a splice site variant, or a frame shift variant, again within the genes of interest. And from that point, I went on to curate those variants. And if you can imagine, actually, for every single one of those participants, there was at least one variant. And so this was many, many hours of work to pull out those which were tier 1. And basically that means they have a recognised association with disease, either because they're protein truncating or because they've been curated by an expert committee to say that. And that took a very long time. And if you imagine that is just for 4 genes, imagine expanding that up beyond 4 genes. So what I'd like to show here, and again this is just part of my experience, is that actually each of these scripts has a differing ability to pull out variants of interest. And so, although I've written sensitivity here, obviously this is quite a crude analysis because actually we don't really know what the true positive rate is. But I think the point that I'm trying to make here is that for each script, they were able to pull out different variants, but actually if you combine the utilisation of all of those scripts, it really maximises variant identification. So the next step in this will now be validating some of the results that I found through visualization in IGV within the research environment, but also comparison with the tier variants which have been produced for some of these participants. So on to results now. So just before I discuss my results in terms of things like CheckIT and PALB2, what I wanted to do is just give a sort of a background slide just so you get an understanding of what we might expect to see with BRCA1 and BRCA2. So the middle column corresponds to the POSH study, and actually what that shows is that if you offer BRCA1 and BRCA2 testing to women who've been diagnosed with breast cancer under the age of 40 years, the detection rate of a BRCA1 or BRCA2 mutation is about 12%. If we move to a study which is much more a population-based study, you can see the detection is much lower, and we could infer that the rate in a familial cohort may be the same, it's probably going to be higher. But I think what I just wanted to show is the percentage detection. And this is what I've seen so far within the GEL cohort. So here, this slide is illustrating the percentage detection rate of TP53, PALB2, CHEK2 and ATM tier 1 variants within the familial cancer cohort. And this, I think, is quite a useful slide and a useful piece of information, because if we think as we're moving on to testing more people, perhaps at the time of cancer diagnosis, one of the key questions is who is the best group to test where we're going to maximise actionable risk identification versus finding variants we can't interpret. And I think this shows really nicely that actually, obviously our detection rate is highest amongst the familial cancer patients, but the detection rate is still reasonable within the under 40s, and then it drops down within the unselected group. CHEK2 is the highest identified gene after BRCA1 and BRCA2, and actually that relationship is maintained through all of the cohorts, followed by PALB2. So now if we focus a little bit in on, well, firstly, I just thought it would be useful to give you a sort of idea of what the overall detection rate may be if we extrapolated what we know from POSH. And it shows that actually the detection rate is pretty good within this familial cohort and also the under 40s. So just some results now. So I next went on to look specifically at CHEK2, and I've compared individuals who have a tier 1 variant in CHEK2, and I've gone to look at their tumour histopathology. And really that was to see whether there are any identifiers within the tumour which may predict the likelihood of having a CHEK2 mutation. And what it shows is that CHEK2 gene carriers are much more likely to have a grade 2 tumour at presentation, but with significantly higher levels of nodal involvement. We can also see that CHEK2 associated tumours are significantly more likely to be estrogen and progesterone receptor positive compared to non-carriers. And this is interesting because actually within the POSH cohort we've been able to correlate the CHEK2 gene carriers against their survival at both 5 and 10 years. And what that is shown is that overall survival is significantly worse in individuals who have a CHEK2 gene alteration at both 5 years and 10 years, and that relationship holds after multivariable analysis. And whilst the numbers may be small, this observation in terms of histopathology and the tendency to higher levels of nodal involvement has been noted by other researchers as well. And so it shows that although this is an ER-positive associated gene, actually this may be implicated in adverse prognosis, and generally ER-positive disease is thought to have a better prognosis. So to summarise, I've talked about genomic testing for hereditary breast cancer. I've given you an overview of my research projects and an experience of my experiences of the research environment at the current time, and some insight into my initial results. I think there have been lots of challenges. I think developing the bioinformatics skills to interrogate the data sets has been challenging, and it's taken some time. I think it would be really, really nice to see if the histopathological associations I've seen within the POSH data set could also be observed within the 100,000 genomes data set, which is a slightly different group of patients. And I hope that that information will become available with time. And I think as well, data management and transfer of information has been difficult. So, as I said, next steps will be looking at tumour histopathology within the 100,000 genomes data set. I'd also like to start looking at some of the somatic mutations. And part of that is recognising that there are specific groups of genes which seem to be more frequently mutated within breast cancer. And actually, if you look to the cancer genome census, you can see lists of genes which are more frequently noted to be mutated within breast cancer. And within that, it's also recognised that specific breast cancer subtypes also seem to have specific gene alterations which are more frequently observed. And I think part of the somatic analysis I'd next like to look at is whether actually specific gene alterations also have particular molecular patterns associated with them, which may help to better understand what we've seen in terms of outcome. Okay, thank you. And I'd like to thank